visiting us UTHealth at Houston. Dr. Chen has a background in biostatistics, and right now he's an associate professor at UTHealth. And his research group focuses on developing statistical methods and computational tools for genome-wide association study and gene-environment interaction studies. And without further ado, I would like Dr. Chen share his research.
Okay, thank you very much, Dr. Yi, for the invitation, for the nice introduction. Today I'm going to talk about some very interesting work in our group, which I call reference panel-free stochastic summary statistics for diverse populations. So there might be some technical details in terms of more complex statistical models, but I think I will try to, share the idea and then look at what's the intuition behind this. So the whole motivation of doing reference panel-free summary statistics is because of, we know that there has been a lot of genome-wide association studies, and the number of individuals in GWAS is approaching, like 1 billion. And this was in 2022. And you can see that most of these individuals were European populations. I think historically, genetic association studies and the GWAS, they have been, widely applied in Europe and also in North America. But we can see that some of the other regions, populations, they are coming up. Very small proportion of the overall individuals available in GWAS. One challenge for analyzing these some of these other populations is because of the genetic background, like Hispanic Latinos in the United States. They are mixed individuals, which means that they don't have a homogeneous genetic background. And their genetic component is actually quite different, and which poses methodological and analytical challenges. And so this is a very brief introduction of the problem of confounding by population structure. This is a fundamental problem in human genetics, because definitely you cannot manipulate their genome. And all the studies are based on observational data, and the confounding is a big problem. So this was one of the examples from 2005, and they were trying to show that population confounding by population structure could happen even within populations in the Europe, because before 2005. People don't believe that confounding by population structure is only going to be a problem when you look at populations that are very divergent from each other. For example, if you combine a whole with Africans, then that's going to be the problem. So here in this study, they found a very interesting example. They were looking at the association between height and some genetic markers. And at that time, they did not have genome-wide data. So they look at some candidate markers. They found very interesting. And the one association, which they showed in panel A, they found that in the population across all Europe, the blue allele and the yellow allele, they had a similar allele frequencies, right? And in this case, you can say that the association is probably true, right? Not confounded by the population structure, because the blue and the yellow alleles, they have approximately the same frequency in all the subpopulations. On the right-hand side, this is very interesting. You can see that the blue alleles are more frequent in Northern European populations, and the yellow allele was more frequent in the Southern European populations. And we also know that there's a gradient in terms of the average height. If you look at the data, people from Northern Europe, they tend to have a higher average height than people from the Southern Europe. Do not take this factor into consideration. And then when you simply look at the association between height and the blue allele, you will see that the blue allele is strongly associated with increased height, right? So this is very intuitive example, but actually it turns out that this was not the case because, for example, if you look at the association between the blue and the yellow allele in Swedish or in Italians, you don't really see the association. So that means, if you take the population structure into consideration, when you account for the population structure, when you account for the fact that these different individuals came from different subpopulations, then you would not see the association between like yellow alleles and height. So, and there's another interesting example, right? So, this was published in Nature in 2008. John November and his colleagues tried to make a plot of the principal components. So basically, these were computed using genome-wide genetic markers. And they were trying to make a plot and see where these individuals are located on the principal component map. It turns out that this map looks very similar to the geographic map of Europe. And you can see that these individuals are clustered around their, the centroid of their, the countries that they came from. And this is very interesting and also justifies use of, ancestry principal components as a surrogate for the population label, because, often have these kind of labels, you only have their genetic data. And then, we can try to make use of these ancestry principal components to control for confounding by population structure. And this, would be a very useful tool to control for confounding by population structure, as long as your study population is heterogeneous, which means that we don't have a uniform genetic ancestry background, right? So you can see this happens even within people from different parts of Europe. And okay, so back to genomic summary statistics. Because many of the studies, they have performed a single variant, genetic association test in genome-wide association studies. They have generated a large number of, in terms of results. And usually these summary statistics are in the form of regression coefficients and their standard errors, or sometimes the z-scores, by dividing the standard errors from the regression coefficient estimates. So these are very rich, resources that's openly available, publicly available, and without having to touch the original individual level data. And they have been used in a wide range of applications. And sometimes certain applications also need the cross-variant correlation, or sometimes, in genetics, people often say that this is a linkage in disequilibrium. Basically, that's correlation between the genetic markers. And, because we don't have this information from the GWAS subset, these are often estimated from external reference. And sometimes they are estimated from the study samples if you have the individual level data available. But the challenge is that, because genomic summary statistics have been applied in a wide range of applications, many of these applications, such as heritability, coheritability estimation, conditional association test, variant set test, LD score regression, multi-phenotype analysis, or transcriptomic wide association analysis, or polygenic risk score. many of these applications will require the knowledge of linkage in disequilibrium between the genetic markers. So, this is really a significant problem. And oftentimes people tend to estimate these from internal reference panels, but this is not going to be a good approach if you have very heterogeneous populations. Following slides. So basically the limitation of these traditional genomic summary statistics is that genetic variations, the single variant summary must be combined with these covariance matrices in meta-analysis if you want to combine them together, because usually you don't have enough statistical power to test the association between your phenotype and some rare genetic variations. So you have to aggregate them based on some genes or genomic regions, or sometimes based on their physical position, or some other kind of grouping units in a meta-analysis combined with other studies. And all these tools, they some matrices for the single variant summary statistics, and they are usually estimated from the study samples. And in this kind of test, the variant sets must be predefined. For example, you can tell the program, okay, I want to do a gene-based analysis, right? I want to test the association with each of the 20,000 human genes. You can do that, or you can do like a 2 kb, 4 kb sliding window analysis on the whole genome. So you have to tell the program what kind of analysis you want to run, and then you would go to the individual level data and then try to compute these covariance matrices based on the definition, how you group these variants together, right? So you have to have this in mind, and let's say if you change your mind, right? For example, if you have a sliding window analysis results, of 4 kb, and you feel that, okay, so the resolution is not very good. I want to go to test a gene, right? I want to switch my analytical strategy. I want to test a gene-based association. Unfortunately, you cannot use those information to do that, because your sliding window, they have very specific blocks of LD matrices, which does not really fully cover the variants that would otherwise be grouped in a gene. So you have to go back to the individual level data and do it again. So you can see that this kind of procedure is very cumbersome, especially if you want to collaborate, right? You want to collaborate with studies in Europe. They are protected by their, GDPR. They cannot share their individual level data with you. So it turns out that, each study would have to run their own analysis and the summary statistics. But, if you want to change your analysis plan, right? For example, after running a multinational collaboration project, running sliding window, you decide that, okay, I want to switch back to gene-based analysis. Then you have to go to each of these studies, participating studies, asking them, hey, now we have an updated analysis plan. We have to run the analysis using gene-based, we have to use a new way to group the variants. Then those guys have to go back and then run their analysis using individual level data again to get the covariance matrix. Because, if your covariance matrix does not cover that group, then it's not useful. So everybody has to go back. So it will take, collaboration very, very inefficient in terms of the whole genome sequence data analysis. Even though you have the data, I have the data, they have the data, you cannot logistically combine them together because of legal restrictions, but then you still want to collaborate. This is very complicated logistically, right? Because you have to harmonize the genetic variations first, and then decide how you want to group the variants. If you want to make slight changes to your analysis plan, you have to start over. It takes several years for you to really work together on something, without sharing individual level data. And also, computationally expensive if you have a large covariance matrices. In this type of applications, LD matrices from external reference panels are not very useful, because when you look at the rare variants, some of them may not even exist in the reference panel you're looking at. And the LD the linkage disequilibrium information is quite different depending on which data set you're looking at, right? And another challenge is for not only for rare variants, but also for common variants. If you have a mixed populations or isolated populations, for example, like people from rural Quebec in Canada, some of those populations, they have been living in their villages for centuries. They are considered isolated, and they're genetically they're slightly different from the rest of the Canadian population. And also, when you look at the mixed populations like Hispanic, Latino Americans, they have very diverse genetic background, so that it's usually very difficult to find external reference panels that correctly represent their LD structure. And if you do some compromise and use the publicly available European reference panels on these populations, you will have reduced accuracy or performance of these genomic summary statistics-based applications. And another challenge is that when you try to deal with kind of relatedness in the study population, you need complex statistical models like linear and logistic effect. They're often needed to handle relatedness in the study populations, but these are very computationally expensive. So I'm trying to deal with using one uniform framework to the idea behind this is pretty straightforward, but the rationale here is that this is a study from the Hispanic community health study, and you can see that these are the genetic background of study participants. So there are basically 6 Hispanic groups, and here each column shows the ancestry component of each individual, and they rank them based on ascending order of their European ancestry proportion. So you can see that, for example, in Mexicans, you can see individuals with almost 100% European ancestry on the right side, right? Compared to individuals who are almost 100% American ancestry. And these are the Native American ancestry that they had in their genome. So you can see it's not really a homogeneous population in any sense, right? So even though they identify themselves as Mexicans, and you can see this kind of genetic diversity. What's even more interesting is that even if you look at 2 individuals, right, with almost the same proportion of European ancestry, but when you look at a specific genome, a specific genomic region, they could be very different as well because of the nature, 15 to 20 generations ago, because of the immigration from Europe to the America and the mixture, and they have very short segments in their genome that they may inherit from their European ancestor or their American ancestor, right? So you can see this kind of genetic diversity, and if you look at the ancestry principal components, you will see a very nice triangular continuum on the map. There's no way that you can draw a line and distinguish one group of groups. So this is very different when you look at the ancestry principal components of Europeans versus Africans, right? So you can see a cluster of Europeans here, another cluster. You can kind of have those kind of labels, and then you can decide, okay, so these individuals might be confounding my analysis that I have to work on a homogeneous population. But you can see here, you don't have that kind of choice. There's no clear line that you can draw to do kind of clustering analysis to separate these 6 groups. And you can also see that the top 5 ancestry principal components, they are very informative in terms of showing their self-reported ethnicity. But starting from the later, principal components, you will basically see some kind of random noise, right? So this is a very complex study, and with very heterogeneous genetic background. So I think this is a perfect example of why we need those ALD measures from the study samples themselves, because you cannot really find representative external reference panels for this particular study. And each Hispanic background group is different. Okay, so now I want to introduce my idea. So we're going to go stochastic, which means that we're using some randomized algorithms to represent the cross-variant correlation in the study samples. So that these studies are very complicated, and external reference panels would not work very well. Then I hope I will convince you that it's much better to use the LD measure from the study sample themselves for downstream genomic summary statistics-based applications. Okay, and so by going stochastic, we can make many of the summary statistics applicable to the mixed or isolated populations. This is because we're not trying to invent some new methods, but many of the new the existing methods, they were previously not applicable to these more complex populations because you don't really have a good measure of their LD. Once you have a good measure of their LD, you can do a lot of things, right? So for example, we can test the distant rare variant sets. I will show you an example later. And we can also do LD score regression to estimate the heritability and the genetic correlation. So this application also requires you to have some knowledge on the LD between the genetic variations. And I would argue that the best LD would come from the study samples themselves. Okay, so this is a traditional genomic summary statistics workflow. In human genetics, so we would first start with some phenotypes, covariates, and kinship matrix. We would fit a model. This model does not include any genotypes from whole genome sequence data from the null model. And then we need to decide how we want to group the variants, right? Because the rare variants are analyzed, you often analyze in the group. Okay, once we have the group information and the raw genotype data, and from the null model, we can calculate the single variant scores and the covariance matrices. And by combining single variant scores and the covariance matrices from other studies, we can do a meta-analysis. Or we can also calculate LD scores from an external reference panel and then do LD score regression on these single variant scores. So we can do a lot of things. We can do the single variant test, the variant set test for the rare variants, gene environment interaction test, using this information. But you can see this is not very efficient, right? Which means, as I explained earlier, if you change your mind, if you want to change the variant set definition, then everything has to go back to the individual level, data level, and you have to run the whole genome data analysis again. So our proposed change to the framework is that, okay, I still start from the phenotype covariant kinship matrix. I got a null model. From the null model and the whole genome sequence genotypes, I directly calculate single variant score and some kind of stochastic summary statistics. So I do not need the variant set definition until the very end stage, right? So before that, I would generate some random vectors from the null model and multiply the random vectors with the whole genome sequence genotypes to get the stochastic summary statistics. And then by combining these information from other studies, I can do a meta-analysis. And I only need the variant set definition at the meta-analysis stage. So you can see that these random vectors are the key for these applications. And also, if you want to do LD score regression, you don't need an external reference panel. You can generate the random vectors directly from the whole genome sequence genotypes to calculate the LD score. And because we move the variant set definition here, so this will really make a collaboration easier because every study would have to just run their analysis without any knowledge on how you would want to group the variants later, right? So you could generate the single variant scores and the summary statistics, without having to know how you want to group the variants together later. And once you receive these these statistics from each and every study in the meta-analysis, then you can decide, okay, I want to do a gene-based analysis to it, right? I want to do a sliding window analysis, using the information. I don't have to go back to each individual level study, ask them to rerun. I want to test some crazy hypothesis. I want to look at these variants on chromosome number 1, along with some other variant on chromosome number 6. I can still do that, at the summary statistics level, and this will really make it very computationally feasible. Okay, so some statistic background. So we consider a generalized linear mix effect model. So basically, this is a model with some covariate and genotype marker, and we have a random intercept to model the correlation between the observations. So this is a very general framework with some modifications. For example, if you don't have the random events, right? This would become a generalized linear model, which include both linear regression and the logistic regression. If you have a logical function, then this is a logistic regression, right? And if you have an identity link function and a continuous traits, then this is a linear mix effect model. So this is a more general form, and that covers a wide range of applications. No matter what you have, we would first fit a null model without any genotypes. And this is what we have. And you can see that this model does not depend on your genotype marker. This is the null model that I mentioned before, right? So I don't need any genotypes to fit this model, and I only need to fit this model once. Even though this is a very complex, I only need to do this once in a genotype association study. Once we have that, then we can calculate some score statistics by looking at the cross product of your genotype markers and the residuals from this null model. And we can also calculate the variance of these single variance statistics by calculating. So this is like a quadratic form. We have a projection matrix in the middle. So in the interest of time, I'm not going to the details of how to compute this projection matrix as a very simple, form. When you want to test multiple variants together, for example, like in the variant test, sequence kernel association test, which is a variance component test of multiple genetic variations. So we could use a quadratic form of the single variant score with some weight matrices in the middle, and then we can compute the p-values by looking at the eigenvalues from this matrix. So it's a little more technical, but basically you can see that the information we need are the single variant scores, right? And these, covariance matrix, G transpose PG. The challenge here is that this matrix needs to be computed using individual level data. So if you have dense genetic relationship matrices, this calculation scales quadratically with your sample size because your projection matrix P here is an n by n matrix. And for rare variant association tests such as SCAT, you need to predefine how you want to group the variance G. Because once you define G, then you can calculate this V, right? Otherwise, you cannot do this genome-wide, and because you're going to end up with a very large matrix, and the matrix is not on full rank. Okay, so here's the idea. When you have the genotype data and the projection matrix, which came from the null model, you multiply them together, you get this V matrix. What we propose to do is to use length and random vectors to represent the information in P, and then we will assemble them into an n by B matrix R. And this B is a number of replicates vectors. In practice, we use 1,000 and found it work extremely well. So if you think about large studies like UGA Biobank or all of us with hundreds of thousands of individuals, this could really reduce the dimension and also the computational complexity. So what we have here is that instead of computing this V matrix, we compute a cross product of the genotype and the random matrix R. So we get this U matrix. And you can see that by the law of large numbers, if the number of variants you're looking at M is smaller than B, when B goes to infinity, you will see that this cross product will converge to V, right? This is true if your B goes to infinity. In finite samples, we don't really have that. But we're using the square of the singular values of U divided by B because you can see this matrix, right? This half matrix, basically. We have the we do a singular value decomposition, then we can get the eigenvalues of this V, provided that B is greater than M. If you don't have that, you can still do the calculation that, without specifying how we want to group G, we can still compute U for the whole chromosome or even the whole genome, where you have the number of variants, which is much, much larger than the number of, random vectors be used in your calculation. We can still do that. And you can see that this matrix multiplication has a complexity that scales linearly with both the bits and your sample size, no matter how complex your statistical model is, right? I'm just talking about the generalized linear mix effect model, but you can have even more complex model, like a generalized additive mix model with some kind of splice effect. You still have a linear complexity in terms of this calculation. So one caveat is that, okay, so now my this matrix does not converge to V anymore if your M is much greater than B, right? So it turns out that even though this is no longer true, the pairwise LD still holds, which means that I want to look at the LD, look up the LD information between variant I and the variant J. I do not really need the full V matrix, right? All I need to do is to go to those two lines in the U matrix and then calculate the cross product of those two lines. And you can see that this pairwise convergence is still true because you're only looking at two variants together and divided by B because your 2 is much less than B. So you still kind of relationship. So this U matrix contains a lot of useful information. You can reconstruct the pairwise LD for any pairs of genetic variations. It does not even on the same chromosome, as I mentioned, right? So, or, very far away from the chromosome. Traditionally, you don't really have that kind of information, or you are not interested in computing the LD between them. But theoretically, using this kind of representation, you can easily compute this LD for very far away variants. And here's the idea, when you have the variant set definition, traditionally you have to define how you want to group the variants together. And, when you change the definition, you have to go over all the way back to the individual level data. There are some recent publications which use, for example, like a banded sliding window approach. They use a 500 kb sliding window so that you're covered as long as you're looking at variants within 5 kb from each other. This partly solves the problem, but when you look at variants that are farther away from each other, you still do not have that, right? And the second approach is to use external reference panel. And for rare variants, they do not even exist in the reference panel. So at the end of the day, you get a very, strange matrix that, you may not even have the same number of genetic variations. And stochastic summary statistics, we can calculate this U matrix first. And then now I can decide how I want to group them, right? I want to color them different colors, and I just look up the corresponding rows. So this is very useful, and we can define how we want to group them later. And this information, LD information is retained for arbitrary variant pairs. So it's not really restricted to regions that are in close proximity of each other. So you can now look at long-range LD, for example, right? And now you want to change the definition, have some strange ways of grouping. You have these different colors, you have to go back to individual level data. And if you use external reference panel, it's not working well. But now we only need to change how we color this U matrix, right? We don't have to go back to individual level data. So if we share this information, you can do a lot of things. A lot of downstream application can be applied to these stochastic summary statistics. And I will just show you 2 examples in the interest of time. So in this case, we were trying to group the variants using a very strange way. We are looking at some high chip data to identify chromatin interaction regions, and some of these interaction loops are close to each other, but some of them are farther away. So you don't really have the regular sliding window approach. You would not be able to cover the LD information between this group and that group. But you can see that using stochastic summary statistics, when we jointly test them together, we get a p-value, a small p-value here. But if you look at these 2 regions separately, you don't really have that kind of significant p-value. So, and we don't have to go back to the individual data. This has all been performed after we have the stochastic summary statistics from the whole genome sequencing study. And you can see that we have a very complex statistical model to account for 3 different types of random effects from the linear mixed effect model. This is very complicated because we need these multiple random effects to account for the study design and the correlation, because this was a community-based cohort. And, and we can see that when we use the stochastic summary statistics, we can dramatically save the time and memory footprint compared to directly analyzing individual level data. So these are the numbers that we analyze on each chromosome, and you can see dramatic saving. And in terms of the accuracy of the p-values, that there's very high correlation in terms of the, this is a Spearman rank correlation. And, so that's the same if you look at the rank correlation of the p-values and the minus log 10 transform the p-values. So they're the same. Okay. So you can see this is very accurate and saves a lot of time and computational resource. Another example I want to talk about is how to calculate the LD scores from the genotypes. And here you don't need the phenotype data anymore because the LD scores are directly computed using the genotype data. So traditionally, we would have to decide how we want to, what kind of flanking region, right, we want to use in the LD score calculation. So usually this is like a 1 million base pair or something like that, plus or minus 1 region, and then you would take the square of the LD and then add the of the LD score. That's how you can do it. If you do this from an external reference panel, unfortunately, some of the variants, they may be missing or they may not be common, right? So because this approach are only applied to common variants, if they are not common, if they are less than 1% binary frequency, then they are removed. They do not exist in your external reference panel. So you get this broken LD score. You're missing some variants, some other variants. You don't have the variants in the data, and these are not very accurate. I will show you one example. And in our framework, we can calculate this U matrix first, and then we can basically extract those corresponding rows. And remember, this is not a large matrix anymore. This dimension B is really just 1,000. So we have 1,000 replicates of these, and then we can go to the data and extract corresponding rows efficiently, and then do the calculation, on the fly. And so we compared LD scores from the study samples and the external reference panels using 3 studies. I mean, basically 2 studies. Hispanic community health study, we have Hispanic Latinos, and we have another study called atherosclerosis risk in community study, the ARIC study. They had whites and blacks. So we look at these 3 groups of individuals, and we compared them with a publicly available reference panel from the 1,000 Genomes project. we look at populations, African American, and then all the different combinations of these reference panels. And we found something very interesting. So first of all, we look at the Latinos, right? This is a comparison between running LDSC, the LD score regression, on the study samples. So we have individuals. And on the y-axis, we're showing the LD scores computed from using the stochastic summary statistics. We can see these are highly consistent, accordant with high Spearman rank correlation. And you can see other 7 panels. These are the different combinations of external reference panels. Not surprisingly, you can see that none of them perform well, right? So they have much worse in terms of correlation computed from the study samples at the gold standard compared to the LD score compute using external reference panels. So you can see none of them work as well as using the study samples themselves. This is Hispanic samples. And if you compare the computing speed, you can see here the purple line shows the LD score regression on the study sample. And for the longer chromosomes, it could take 2 months if you are running them on a single thread, right? Of course, you can do threading to bring down this to maybe 1 or 2 days, but you still need more than 2 CPU months to do the your sample size is 7,000. If you're trying to think about applying this to all vast data with over 400,000 individuals, it's going to explode. Okay, and using our stochastic summary statistics, you can see very low computing requirement. It's even faster than running LDSC on the European superpopulations from 1,000 genome. And I think these, only include a few hundred individuals here, right? So you can see that we can save a lot of time. And in terms of the heritability estimates from LD score regression using LD scores estimated from these different methods, you can see that our stochastic summary statistics always give very similar heritability estimates compared to the gold standard using LD score regression on the study samples. The other combinations of reference panels resulted in kind of bias in some of these phenotypes. Okay, but the limitation of this study is that because we only had 7,000 individuals, so these heritability estimates themselves may not be very accurate because you typically need a much larger samples to get a better heritability estimates. But what I'm trying to show here is that even if these heritability estimates are not very accurate, we can see a very good consistency between the randomized algorithm and the deterministic algorithm, which would take several months. Okay. And yeah, here's a take-home message. Stochastic summary statistics are more accurate than those from external reference panels. And also, it's the most computationally efficient. And here's another example from the white populations in ARIC. And you can see here, the first plot shows a comparison between LD score regression and the randomized algorithm using stochastic summary statistics. You can see very good consistency. And you can also see good consistency if you're looking at 1,000 genome external reference panels, right? Using the European superpopulation, you still get a very high correlation of 0.98. So the other reference panels did not really work very well, but you can see this is the benchmark because a lot of people are doing this right now. And you can see this is a justification, right? So if you have whites using external reference, very bad idea. It works very well. And this justifies the choice, right? When you look at the heritability estimates, when you compare the third column with the first one, you can see that they're pretty consistent, right? So that means, in this particular case, using European populations from external panel, reference panel performed reasonably well. So that's a justification why many external reference panel-based summary statistic approaches have been widely used, but they're only restricted to studies with European ancestry participants because they work and they work well. If you look at the blacks, and we know that these are basically mixed individuals with both European and African ancestry. Again, you can see good concordance, using the randomized algorithm. But in this case, when you look at the European population from 1,000 genome and the reference panel and the African population, you can see some kind of reduced performance. And also when you combine them together, right? So you kind of see this discrepancy and also this kind here and here. So this is because these individuals are admixed. And even if you can find reference populations that represent their ancestry background at the local level, because LD score are local measure, you may have still very different LD, in terms between your study sample and your reference population. And remember that Africa is a continent with the largest genetic diversity. So the African individuals in the 1,000 genomes, they may not be representative of the African ancestry in a particular study, right? So here you can see, very dramatic difference. Again, the study sample is very small. I think we only had like 1,000 individuals, but you can see huge difference. And in this case, none of the external reference panels performed well. Even if you combine them together, right? You know that African ancestry plus European ancestry didn't represent the ancestry component of my study samples. But in this case, you can see, even if you combine them together, look at the local ancestry or, the linkage disequilibrium of particular genetic variations, none of these external reference panels perform well. So basically, I think our conclusion is that using external reference panel is generally not a good idea for mixed populations, not only for Hispanics, but also for Blacks in our study. And even if the reference panel covers all ancestry populations, you still do not have good performance. And we also tried calculating the genetic correlation between these pairs of phenotypes in Hispanic community health study. And you can see in all these cases, our randomized algorithm, stochastic summary statistics-based approach gave very close genetic correlation estimates compared to the LD score regression on the study samples. So here are the take-home messages that I to summarize my talk today. Stochastic summary statistics, is a novel computationally efficient framework for using to represent cross-variant LD structure from whole genome sequencing studies. And the computational complexity always scales linearly with both the sample set and number of variants, regardless of how complex your statistical model is. You only need the complex model once in a genome-wide study. That's your null model. And all the calculations are basically matrix multiplications. So that's why it scales linearly. And also we can allow for flexible variant set definitions for under-investigated scientific hypotheses using this framework. I can look at a long-range LD or how to group these variants together. So previously, the long-range LD information is not really richly available, and now we can make them available. And we can also enable flexible meta-analysis to facilitate international collaborations on whole genome sequencing studies. For example, if you want to combine UK Biobank and all of us data, you cannot do that because UK Biobank, they have their data hosted on one cloud computing platform, and all of us, their data are hosted on another cloud computing platform. Not allowed to download and combine their data. You are not allowed to move data from one platform to another, but you can combine their summary statistics, right? So, with this kind of tool, you have much more information that you can combine from the two studies. You don't really need to decide how you want to group your variance. The summary statistics and will combine it together. And this will really facilitate international collaborations because, the data are subject to jurisdictions and they have different laws, and you cannot share individual level data, but you can share summary statistics that can still facilitate collaboration. And there's really no need to use external reference panels. That's what I'm advocating today. So because using these external reference panels are not very good for a mixed and isolated populations. And if we're dealing with a more heterogeneous population, such as the All of Us study, right? You have a lot of individuals from very diverse ancestry background groups, and you don't really have good reference panel that represent LD between those genetic variations. And at the end of the presentation, I would like to thank my team and my collaborators, and also Eric and the SOUL study participants, the Transomics for Precision Medicine program, and funding support from the NIH. I'm happy to take any questions.
I saw there's a dramatic differences in the heritability estimation across different populations. My question is, do you expect there's a fixed, for example, expected heritability for LDL, and then why?
Yeah, I would suggest not to overinterpret these results because our sample sizes are very small. So we're just using this as a proof of concept. So we were trying to show that our heritability estimates are close to the gold standard, which is the in-sample LD combined with the summary statistics from the study sample themselves. I don't have any confidence in saying that these heritability estimates are accurate in any sense because the sample size was very small compared to, you can get probably more reasonable estimates. That's what we're doing in All of Us right now. So we have over 400,000 individuals. You can get much more meaningful heritability estimates from those studies, but unfortunately, I don't have those results yet. But this was a proof of concept. Yeah. So you definitely have some kind of bias when you try to estimate heritability using only a few thousand individuals. So I would suggest not to overinterpret these results.
I know that LDSC just give you the lower bound heritability, and it seems that your method is still a little bit lower than LDSC.
I would not say that this is lower.
Remember, for Latino populations, right? So do you have any idea to improve those heritability estimation? Is that
I think the interest here is not to show the heritability, right? And you can see these are within the error bar. I would not say that they're lower. I think they're equivalent to each other.
To some statistical aspects, right, very naive ones. So when you generate those random vectors, does it matter how you generate them?
Okay, maybe. Sorry. So I skipped the technical details. So basically, these were simulated from multivariate normal distribution, and the purpose is not to represent the p-matrix. We know that this is a dense matrix, but what we're arguing is that the cross product of these random vectors, each of these random vectors, can represent the variance or covariance matrix of this G transpose PG, because this is a dense matrix. This is if you look at one specific group of variants, this is considered to be a small matrix. Compared to your sample size, right? Or, the number of, replicates your random vectors. So, that's the law of large numbers. That, the idea is very, very, very naive. I mean, but I think, showing is that this is really useful for people, especially when people try to look at some longer range LD. You don't really need to define how you want to compute the LD or define the variant group, and you can still preserve those information. Even you apply this technically, we're applying this whole chromosome, computing this once for each chromosome. But in reality, you can get a lot of information by doing some lookups. If you want LD information from arbitrarily varying pairs, you can still have that.
I have an even more naive question. What's the matrix P here?
Oh, that's a projection from the complex statistical model. So this matrix will depend on the model. So if it's a simple linear regression, this is a very straightforward. It's gonna be an idempotent matrix by projecting out your covariance effects. But for mixed effect models, this is complicated. But it's a dense matrix. Yeah, it's an n by n matrix. So that's why we try to avoid making those kind of computations.
You said B, that you take B equal to 1000.
Yeah, that's
that
empirically. That's
with
what
that size. If you throw in another random values, how much variation are you going to see in your final results?
Yeah, that's a good question. So we tried this, using different numbers, in our research. We tried 10, 100, 1000, and all the way to 10,000. We saw 1000 was recommended because it's a good balance. Of course, if your dimension gets larger and larger, then your computation will also scale linearly with the number of B, right? And also, the kind of storage you have is also scaling linearly with your B. By using smaller number of B's, for example, like a 10 or 100, it's good enough if you're looking at a single variant association GWAS, but not for their variance or LD score calculation. 1000 works well in this case. 10,000 is much because you're paying the price of computing, and then yeah, I think that's a good balance. I did not show the comparison here in the presentation today, but that's the technical experiments we tried.
So B does depend on your data pattern, also depend on the big N as well.
It doesn't really depend on the sample size. That's a very good question. So it depends on how many variants you want to look at together. So let's say if you are running rare variant analysis and most of your variants are, a few hundred variants together, right? 1000 is good enough. But if regions, let's say if you want to test 10,000 variants a time in a sliding weight analysis, usually people would not do that because your resolution is low, if you group a large number of variants together. But if there's such a need, then 1000 is probably not enough to represent, because you can only get 1000 singular values from a singular value decomposition of this matrix. You can get at most 1000. And all the remaining ones will be assumed to be 0. So that's going to be a problem. If your V is a, let's say, 10,000 by 10,000 matrix, you can get the leading 1000, but then the remaining ones will be 0. So you can underestimate your p-value.
So intuitively, which component give you some stochasticness in the estimate later?
Is R.
The R.
Yeah. Because this is a completely random matrix generated from P. You're simulating this R, some multivariate normal distribution. Yeah.
And what parameter will impact the accuracy of the final estimate? The number.
It depends on the application, the downstream application. For example, if you're interested in estimating heritability, right? So basically, B will have an effect. Definitely, if you have a larger P, then you have more accuracy. And also it depends on the window size you want to look at, right? If you're doing like 1 million base pair flanking region, usually you will have several thousand variants in those regions. And yeah, I think in terms of LD score, it's still good enough because LD score is still a single variant level measure, even though you're including a lot. Because as long as this is accurate, then the accuracy is still bounded. But you know, if you try to do yeah, but they add up together, a large number of them, you could still see at some point they will not be good enough.
Yeah. So because I was looking at you're testing the in sample one from LDSC and then the one estimated by your method.
Right.
And you
So I
can,
think some of the variability came from the fact that the LD and then take the square of many variants together, right? So for each one of them, you have this kind of pairwise convergence. But when you add tens of thousands of them together, you will see some kind of noise. Yeah, okay, right.
cool, we're actually over time. Thank you.
But if you're trying to like